So we were talking about the structure of the chromosomes and in particular the telomere structures that we can see here. So the telomeres, you can see them here painted again with this fish technique that we will discuss, are the way that the chromosomes are kept separated in the cells. They're very important, of course. They are the only reason why we can keep the different chromosomes separated. And but how to close, how to make a loop, you know, at the end, enclosing the DNA. That's not completely easy because, of course, you always have the problem of the 5', 3', 3', 5' orientation in the DNA. So the way it is done is that normally you see this is 5', 3' strand. This is the 3', 5' strand. You create the loop in this way. You have a number of proteins, TRF1, TRF2, TIM2, RAP, TANK, SPOT, and other proteins that allows this kind of loop in the DNA. And at the very end, you leave a 3' hanging length here so that you can actually close the loop. But what will happen is that every time there is a division, this hanging part will become shorter. This we will see. Why telomeres are important? I mean, telomeres are important, you see, because when you break the DNA with chemicals, or of course, in our case with radiation, so when you induce a damage into DNA, the cell has to react. And this is the broken DNA, let's say. If you don't have telomeres, there will be no way to distinguish, let's say, the normal status of the cell from the broken chromosomes. Moreover, and that's of course, if you lose the telomeres, what will happen is that the different chromosomes will start to fuse. Essentially, they are rejoined, as happens when you have a break into the DNA induced by ionizing radiation. When this happens, you have these situations, you see, and then you create a bridge. This bridge can be broken in mitosis, but then the DNA is now mixed, is broken in different pieces, and you enter is what is called fusion bridge breakage cycles that are the typical hallmark of genomic instability. Essentially, the moment that the telomeres become so short that there is no more division between the different chromosomes, this status is not compatible with the life of an organism, because at this point you will fuse your DNA and you will enter in total instability of the genome. And this is interesting for the reason I was telling you before, because you see, you always have this 5', 3', 3', 5'. Now, DNA replication is bidirectional, but the polymerase always move. We have seen this already for the DNA replication. The polymerase always move in 5', 3' direction. So what happens? It happens, as you see, this is going 5', 3', and this is going now 5', 3'. So what you need a primer where the polymerase can actually attach. So when you are at the end of the chromosome, you will end up with this prime, but every time you have a replication, you then leave some 50 to 100 base pairs of DNA unreplicated at the 3' end. So the legging of this part is longer, and the chromosome is shorter now. So the telomeres are shortened; they become smaller and smaller and smaller until eventually you go in the senescence. So this is the link between telomere length and life, if you want, aging in general, is what is called the Hayflick limit, because it was first introduced by Hayflick, who was one of the discoverers of the telomeres. And you see, essentially, what happens is that in normal somatic cells, you start with the nice long telomeres, but then with age, this, and with the number of doublings, of course, because this happens every time you double the cells, these telomeres become shorter and shorter and shorter until the cell will be unable to replicate anymore. You go in genomic instability, there are no more chromosomes, it's fused altogether. Two alternatives to this, not very good, is one is, of course, the germ cells. These are telomere positive, so they can keep replicating, but keep in mind that, for example, for the eggs, this only happens at the beginning, and then they stop completely. But for other germ cells, like for the sperms, they can keep replicating all the time. The other alternative is that you have a telomerase, so an enzyme that makes the telomere always longer. So add the piece at the end of the telomere and makes it longer. This is apparently good, but in reality, many tumors use telomerase to escape senescence that would be for them, of course, the end. So the telomere hypothesis of aging is quite controversial. First of all, the telomere length is not related to the lifespan. So if you take the mice have much longer telomeres than humans, but they live much shorter. And even within the mice, there are different species that live the same, but have completely different telomeres. So the point is that telomeres contribute to aging only if senescent cells contribute to aging. So only if you have cells that duplicate, age, those cells contribute to aging. On the other hand, telomerase protects against replicative senescence, but not senescence induced by other causes. So if you have senescence not related to the replication, telomerase are useless. And there are several inducers of cellular senescence that have nothing to do with telomerase. So it's a fascinating idea, and in a way gives you a sort of extreme limit of the aging, because you say, okay, if nothing else happens, at least because of the telomerase shortening, sooner or later, you have to die. And actually, in humans, this limit down here is estimated somewhere around 120 and 130 years, but it's not enough. What is interesting, though, is that you see correlation of the telomere length with several events, including stress. You know, we normally say stress shortens our life. This is an interesting experiment, a mix of molecular biology and psychology, where actually they have, for example, if you take this plot here, this is the telomere length, and this is the perceived stress scale. So these people, these are normal people, they are not patients. They reported in a formula what is the stress that they perceive. And you see that there is a correlation. The shorter telomeres is higher stress. You also see it here. You know, this is the average telomere length, and this is the telomerase units. You see that is lower for the high stress as compared to the low stress. But as I said, I mean, aging is more complicated than telomeres. This is also a nice paper called The Hallmarks of Aging. We will see later that there is a famous paper called The Hallmarks of Cancer. This is the hallmarks of aging. And you see there are many different effects. You have altered intercellular communication, genomic instability, telomere attrition. Attrition is the reduction, is one of the hallmarks of aging. But you have epigenetic alterations, in particular methylation, an increase in methylation. loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, so in energy, of course, cellular senescence in general, which is also related to telomere shortening, stem cell exhaustion, you don't have any more stem cells. Also, this can be related to telomere attrition. The epigenetic alteration, so the methylation, which, as we have seen before, is responsible for the compactation of the chromatin and for the lack of expression of various genes, is so important that actually you can define, based on the degree of methylation of the DNA, you can define what is known as Horvath's clock. So you can actually measure the real age of an individual based on the amount of methylation. You see, this is the weighted methylation average as a function of the age. Okay, in the first months and years of life, methylation increases very quickly, but then it's essentially linear. So this is the average epigenetic age. Now, of course, depending on where you are in this curve, I mean, if you are here, this is the average. If you are down here, you are younger than your actual age. If you are here instead, in terms of methylation, you are older than your chronological age. This is called Horvath's clock. So, going back to the cell cycle, we see that the cell cycle, of course, is the life, the aging of a cell, if you want. We have seen already that you go from G1 to S to G2. What I want to stress now here is that this cell cycle does not happen without passing the checkpoints. There are essentially two main checkpoints in the cell cycle. One is the G1 checkpoint, the G1S, so for the transition between G1 and S, and the other one is the G2M. G1S checks essentially for sufficient nutrients and cell volume. And we will see also for the integrity of the DNA. Also in G2M, of course, you check for the integrity of DNA for the completion of repetition. So if you have these conditions, you can proceed in the cell cycle. Otherwise, the cell is stopped. If it is not stopped, you see here, you have a growth. One way that, and you see this here, one way that people study the cells is very simple. I mean, you take the cells from some tissue and you can grow them actually in bottles or in petri dishes in the lab. And that's how they look at the microscope, how they grow in the microscope, you see. If you measure the number of cells as a function of the number of days here, the growth curve has pretty much this phase. You know, there is a lag phase where there is an adaptation, essentially. Then there is an exponential phase. And finally, there is a plateau because you can see it here. This is, let's say, the exponential growth. But then when they fill all the bottle, they stop, and eventually, if they have not enough nutrients, they die. This phase here, the exponential phase, being exponential, can of course nicely be expressed with an exponential function. So the number of cells will be, and this is obvious, one cell become two, two become four, and so on and so forth. So this will be N0 times 2 to the population doubling number. PDN means population doubling number. And you can write the population doubling number as the ratio T divided by TD, where T is the time and TD is the average division time. Of course, if you like the tau, the exponential, the Napier number, then you can write, and this is more familiar, N as N0 e to T divided by tau. And of course, tau is simply TD divided by the logarithm of 2. It's interesting that you can see already, we will come back to carcinogenesis, you can see already the cancer in vitro, because typically the cancer cells do not stop here. This is the so-called growth inhibition, but they keep growing on top, one on top of each other. And this is a typical characteristic of cancer cells in vitro. For a tissue, the organization of the tissue, essentially you start with the totipotent stem cell. The totipotent stem cells is essentially the first original stem cell that gave rise to us, to a full organism. From the totipotent stem cell, you generate pluripotent stem cells. And from the pluripotent stem cells, you eventually generate the specialized cells. So, to have an organism, you must have stem cells and differentiation of the stem cells. Stem cells are divided in adult, that are stem cells that are now in our body, fetal, that were in the fetus, embryonic, these are really the original stem cells that made the fetus, and then there are also new stem cells. You can now take somatic cells, the somatic cells from your organism, and you can revert it in a stem cell. The characteristics are, of course, they propagate for many cell doublings, they give rise to a wide range of cell types. They are, of course, especially the induced stem cells, are excellent candidates for cell and tissue replacement therapies. Imagine, you know, that you have a heart after an infarct, cells are dead. Imagine that you can take your cell, somatic cell, bring it back as a stem cell, and differentiate in the heart so that it can actually regenerate the heart. It's called regenerative therapy. This is a little bit more on how the embryonic stem cells work. You know, this is the totipotent. The totipotent is essentially what in all genetics is called the zygote. The zygote is the first cell where the sperm meet the egg, and so you have the zygote. Then from the zygote, you know, the totipotent. Totipotent really means that it's able to do everything. You know, from that single cell, you can generate a full organism. You make a morula and then a blastocyte. That now you start to enter in the so-called in the so-called pluripotent stem cells. And from these pluripotent stem cells, you essentially generate three lines: the ectoderm, the mesoderm, and the endoderm. Again, in this ectoderm, mesoderm, and endoderm, you have multipotent cells, progenitor stem cells, and finally the organs. For example, the ectoderm is the brain or the skin. In the mesoderms, you have the muscle and the bone marrow. In the endoderm, you have organs such as liver or pancreas. Now, for what are the adult or somatic stem cells that are, let's say, our own stem cells? Now, they are generally multipotent. They are responsible for tissue homeostasis. There are large numbers for some tissues. So typically, the large number of stem cells that we have are the hematopoietic stem cells, because we need a lot of blood cells. We generate a lot of blood cells. So we need a lot of hematopoietic stem cells. We also have neural stem cells. So we also generate some neurons, but unfortunately not many. So, you know, we don't have so many neural stem cells. A little bit of molecular biology is here. There are typical genes that are involved, are Notch, Wnt, sonic hedgehog pathway, that can switch from what is called the asymmetric or symmetric division, you know, because in the symmetric division of the stem cells, you have the expansion. So if you have a symmetric division, this means that one stem cell becomes two stem cells. When you have the asymmetric division, this means that you now generate something different when you divide, and these are eventually the differentiated cells. So, you know, the hypothesis is that the cells arose from primordial germ cells, migrated outside gonads, and de novo by unknown mechanisms. Oh, sorry. No. Oh, okay. Sorry. This is the example of the blood. You see that this is the pluripotent stem cells that create lymphoid stem cells and myeloid stem cells. And from the myeloid stem cells, for example, you have the erythrocytes, the thrombocytes, but you also generate basophil, eosinophil, neutrophil, monocytes, T cells, and the T and B lymphocytes. These cells are essential for the immune system. This is a very rapidly dividing tissue. We have approximately 10 to the 11 new cells per day in a human adult, which means 10 to the 6 per second. Every second we make 1 million new blood cells. Of course, from the stem cells, we generate structured organs, where, of course, the cell-to-cell communication is extremely important, as shown here, and eventually you organize the tissue. There is one thing which is important in the tissue. It's very important, especially for radiotherapy. We will see it later. I mean, the tissue can be serial, parallel, or mixed type. Examples of serial organs is like in the nerve tissue, you know, the spinal cord, the nerves, these are all serial organs. In a serial organ, also the esophagus is a serial organ, the colon is a serial organ, you know. In these serial organs, if you induce a damage in a point, you destroy the functionality of the organ. But if you have a parallel organ, like the lung, the brain, the liver, in this case, you can damage a part, but the organ is still working. Actually, to be sure, and this you probably already are thinking about that, the brain is not really purely a parallel organ, but it's more a mixed type, because, you know, depending on where you do the damage, it can be irreparable or can be repairable. So some damages in the brain are irreparable. Other types, so they lead at the complete non-functionality of the organ. In other cases, you can have severe damage to the brain, but still it's okay, you can survive. Serial parallel organs are those organs that are, some parts are serial and some parts are parallel, and this is, for example, the heart. The heart is a mixed organ serial parallel. Obviously, you know, in radiotherapy, this is very important, because if you have a parallel organ, if you have a tumor in a parallel organ, you can accept to give a fairly high dose also to the rest of the organ. But if you have a tumor which is close to a serial organ, you have to be very careful, because if you damage the serial organ, also a small volume of the serial organ, that organ will be inactivated. Now, in the final part of the lecture, we will say a few words about cancer. So we have seen already in normal cells, there is a limited division capacity, also coming from the telomere shortening, but cancer are immortal, or almost immortal. Maybe this is an approximation. We will be a little bit of language now. Cancers are divided in benign tumors that are local growth and defined volume. These are not really cancers. These are benign tumors. Cysts, not cancers. Malignant tumors are what we call cancers, and they are characterized by invasive growth and very often metastasis. Not always, but very often metastasis. The main mechanisms we will see is very complex, but let's say the main hypothesis is that the cell transformation is due to mutations, and the genes that play a main role in these mutations are the oncogenes, that are genes that promote cancer, or the tumor suppressor genes that are genes that normally suppress cancer. So the whole story of cancer is a sort of balance. You must have a balance between the stimulating oncogenes and the inhibiting tumor suppressor genes. Both are important. You know, oncogenes generally promote the growth. Tumor suppressor genes generally stop the growth. In a normal organism, you must have a balance. In a disordered organism, this does not happen. One of the most famous tumor suppressor genes is actually a checkpoint gene, and it's called P53. P53, very often called also the guardian of the genome. The P53 plays a key role here in the G1S transitions, because you know every kind of DNA damage activates the P53 gene, which in turns activate the P21. P21 is a kinase, then blocks the phosphorylation that eventually leads to this DNA replication. So it stops DNA replication in G1S. It's a very famous gene. I mean, it was discovered a few years ago. You see, this is the cover of Science of 1993. In 1993, P53 was the molecule of the year, because when they discovered the guardian of the genome, they thought, okay, we have now the key to stop the cancer, because everything is related to P53. Actually, this is very true. Not 100%, but 50% of the cancers have mutations in P53, roughly, of course, roughly 50%. So it's an important step. Inactivate the P53 is one of the main mechanisms for a cancer to become a cancer. But carcinogenesis is a multi-step process. It's not that you inactivate the P53 and you have cancer. As a minimum, you can divide, you will see that there are many steps, but you can identify the steps in three big groups. One is called initiation. In the initiation, you have a modification of a single cell, a mutation, something that somehow stimulates a single cell. This leads to the second step is the promotion. So these mutated cells must be in an environment where it can grow and divide. And finally, the progression, where you accumulate modification and eventually lead to invasiveness and metastasis. The cause of neoplasia are many, and they are, as you know, not completely clear. For sure, there are environmental causes that are related to exposure to chemicals, to virus, or also to radiation. We know for sure that some chemicals, some virus, and some radiation exposure in some cases can induce cancer. But of course, there are hereditary causes, genetic defects, especially for pediatric tumors. They are dominated by heredity, and a combination of a genetic predisposition and environmental causes, or obscure defects. Which is more important? Is the genetics, is the environmental causes? We will see that we have much more information now. In general, this is a study on chemical carcinogenesis. So we know that some chemicals like benzopyrene can cause cancer. They induce DNA damage as initiation phase. If you have inflammation, and this is very important, we will see, we will come back to this one. You have a promotion, and then you see histological change. And then later on, you must have a progression that leads eventually to this malignant transformation. Viral oncogenesis, this is very well characterized, of course. Essentially, you have a virus. We have seen the virus already. Some virus can induce mutations because you have the insertion of the viral nucleic acids in the mammalian genome, leading to alteration of oncogenes or cancer suppressor genes and so on. There are many examples. You see, maybe the most known example is the human papillomavirus. For the human papillomavirus, which is certainly related to the cervical cancer, so it's certainly the main cause of cervical cancer, there is now a vaccine that was developed by Harald Zurausen in Heidelberg not many years ago. He got this Nobel Prize for this. And now the young girls can be vaccinated for human papillomavirus. It's something that started a few years ago, so we still don't know the long-term efficiency of this vaccine, but we will see. Another well-known cancer gene is the Epstein-Barr virus, which is responsible for the Burkitt's lymphoma and for some kind of nasopharyngeal cancer. Actually, also human papillomavirus is very often found in head and neck cancers. And this is, of course, because it's a virus that generally lives in the uterus and in the cervix. It is very often related to sexual transmission if you find it in the head and neck. And finally, hepatitis B and C are correlated to hepatocellular carcinoma. But this is interesting. The real reason why hepatitis is correlated to hepatocellular carcinoma is not the initiation process, like for papillomavirus, but is the inflammation process, because hepatitis induces liver inflammation, so it's responsible for the liver disease. So it's not in the initiation phase, but in the promotion phase. So hereditary cause, here there are so many examples. One typical example is the so-called FAP, family adenopolyposis. This is actually involved in the gene C5 and is one of the main causes of adenocarcinoma of the colon. So colon cancer, if you have FAP, if you have this mutation in the gene C5, very easily you have colon cancer. Retinoblastoma is a rare tumor, certainly related to mutations in the retinoblastoma genes. It's a hereditary tumor. Neuroblastoma is also a very hereditary tumor. And of course, if you have trisomy 21, which is of course hereditary, this is Down syndrome, but the Down syndrome itself is related to a higher risk of cancer, especially leukemia. Leukemia is very common in Down syndrome patients. There is more than a genetic predisposition. The answer is certainly yes. More and more cases are found. In this list, for example, one gene which is missing is BRCA1, which is responsible for breast cancer. And again, the general scheme is that the proto-oncogene becomes an oncogene when you have external influence, including hereditary, radiation, chemical, viral oncogenes or other. The stage when an oncogene is not working as an oncogene, but is a proto-oncogene, so comes before, is working properly, this is why it's called proto-oncogene. Now, this scheme, though, where you have essentially, you have the stem cells, you have a mutation, then you have promotion, initiation, and cancer, is actually a little bit more complicated. In the year 2000, Hanan and Weinberg published this famous paper, The Hallmarks of Cancer. And the hallmarks of cancer, you see, there are several: sustaining proliferative signaling, evading growth suppressors, activated invasion and metastasis, enabling replicative immortality, inducing angiogenesis. Angiogenesis is the growth of capillaries. You need enough blood, otherwise even if you are hypoxic, you need some blood, so you need angiogenesis. You need to create new capillaries if you want to grow. And resisting cell death. These are the so-called hallmarks of cancer. And you see the idea in this paper of Hanan and Weinberg is interesting because they show that cancer is not really a set of cells. Cancer is another tissue. So it's a tissue with its own characteristics. In the 10 years later, in 2011, they published the correction, if you want, to the paper, including two emerging hallmarks. One is the deregulating cellular energetics, and the second one, very important, is avoiding immune destruction, because the immune cells should recognize the cancer and destroy it. Moreover, there are two more enabling characteristics that are genomic instability and mutation. Cancer are definitely characterized by many mutations. The final cancer are genomically unstable. And tumor-promoting inflammation. This is a key, apparently, for the promotion. So you can have many mutations, you can have a lot of mutations, but if you don't have inflammation, this will not bring to cancer. As I said, the tumor, the vision of the tumor as a set of cancer cells attached during the tumor is very simplified. The real world is much more complicated than you see here. I mean, the tumors are tissues where you have cancer stem cells, immune inflammatory cells, invasive cancer cells, endothelial cells, fibroblasts, and so on. And you see that's how you can generate a metastasis from here. So this is called the microenvironment. And these days, targeting the microenvironment turns out to be maybe a best strategy than targeting the tumor. Even though, of course, these cells here, the cancer stem cells, are also the best candidate, because are the cancer stem cells that can generate the other cancer cells. So if we can target the cancer stem cells and destroy the cancer stem cells, then certainly we will have sterilization of the tumor. I told you already that it is a multi-step process. And one of the best ways to see this multi-step process, this is an old paper that came out on Cell in 1990 by Firon and Fogelstein. Fogelstein is still now one of the most greatest experts in molecular biology of cancer. He described already at that time the multi-step model of colorectal tumorigenesis, making a parallel between histological changes in the colon and molecular changes in the colon. So you see, you start with a normal colon, and then the first event that you have is, the first event that you see is a polyp. For this polyp, you have a first genetic event. The loss of APC, which is a tumor suppressor gene on chromosome 5. So you start from normal, then you lose the APC on chromosome 5, and you have a polyp. But a polyp becomes a class 2 adenoma, so grows a little bit, only when you also have activation of KRAS. Now, this is not the tumor suppressor gene. KRAS is a known oncogene on chromosome 12. If you lose another suppressor genes, the DCC on chromosome 18, you have a class 3 adenoma. Then you have to lose the P53. You know, there's nothing to do. At the end, you have to lose the P53, and then you start to have a malignant carcinoma at this stage. The malignant carcinoma will metastasize after further changes, molecular changes that occur. But you see that this was the first example of a parallel between histological changes and molecular changes. So apparently it's a matter of accumulation of mutations. You see, in this model, in the model of Fogelstein, cancer is the result of a gradual accumulation of mutations. There is no promotion and there is no inflammation here, if you want. So this is partly true, but probably not completely true, and we will see in a second why. Again, from the Fogelstein group, more recently, he tried to use this model to answer the original question. I mean, how much of the cancer is due to environmental causes, and how much is related to genetics. So he had the simple idea. He said, okay, when do we make mistakes? When we can induce naturally mutations in our cell? When the cell divides. So we have, let's compare those tissues that have many divisions, actively dividing tissues that are here. You see total stem cells division, high number of total stem cell division. Let's compare them with the cells that have very little divisions, like the bones, here is where they are, the bones, or the brain. This is the risk of cancer, and this is the total stem cell division. Now, if you put the different tumors here, you see that the trend is not exactly aligned, but the trend is that those tissues that have a lot of stem cell divisions, like the colon here, have a much higher risk than those tissues where you have very little divisions, like the bone down here. So what is the conclusion? The conclusion is that cancer is a matter of only of chance, is only luck, is only bad luck. I mean, you divide and you induce mutations, and then eventually you induce the right mutation, and then that will be cancer. Well, of course, not exactly. You have to be careful, because the impact of the environment is also pretty clear here. I give you an example. This is the risk of cancer in smokers. This is the risk of cancer in non-smokers. There is an order of magnitude difference. It's not a small difference. I mean, this is 10 to minus 1. This is 10 to minus 2, or even 10 to minus 3. It's more than one order of magnitude difference. So you see, smoking, so a bad habit, increase your risk of cancer of more than a factor of 10. But still, if you don't smoke, your risk of cancer, for example, will be higher than the risk of osteosarcoma, which is a bone tumor, simply because the bones divide less than the lung. And if you go up here, for example, we have seen before the FAP, the family adenopolyposis. If you compare the risk of colorectal cancer here, less than 10 to minus 1, with the risk if you have FAP or Lynch is another genetic predisposition, you see that there is really an order of magnitude difference. So make no mistake, this is a very interesting plot, but this does not mean that having bad habits, like smoking, for example, is not a risk for cancer. It is a risk of cancer. So environmental exposures are important. But if you look at this one, their the overall contribution, of course, not specific. This is also a mistake. I mean, not specific. For lung is a factor of 10, but overall the contribution is approximately 1/3 due to environment, and the rest is essentially bad luck. So it's essentially simply that you have more division, and during this division, sooner or later, bad luck also means aging, of course, sooner or later you will get the cancer. However, you see there are some paradox. One known paradox is the so-called Pareto paradox. Pareto paradox is about the elephants. So cancer is an aging disease, we have seen, and is related to the number of division of stem cells. So if you have many stem cells, you are big, you have many stem cells, big animal, and you live very long, like the elephants that normally live 100 years or more, you should get many cancers. But this is not the case. In fact, the elephants essentially do not get cancer. And this is also true for other large animals, we will see. One of the reasons for elephant was recently discovered in 2015. They found out that the elephants have approximately 20 copies of the p53 gene. And you remember the guardian of the genome. So while we only have 2 copies of the p53 genes, they have 20. So clearly, they can suppress any cancer much better than we can. It's probably a mechanism of defense. I mean, if you want to be big and you want to live longer, you must have a lot of copies of p53. But this does not mean, and this is interesting, this does not mean that, say, why don't we increase, let's say, the quantity of p53 in our body? Why don't we enhance the expression of p53? Because if you do this, and has been done, you see, there is a paper from 2002 in Nature. These people actually from the Baylor College of Medicine in Texas, in collaboration with the Massachusetts University in US, they actually induced p53 mutant mice that overexpress p53. The result, you know, this is a normal mouse. This is the mutant. Is that this animal actually really has a much lower incidence of cancer, but ages much quickly. So the aging is much quickly, which in a way makes sense because p53 will induce the death of the cells more quickly, so you lose your cells, and then of course it's like aging, you know, your cells die and you age quickly. So there must be something different in the elephant to escape this mechanism, because they don't age quickly, they live very long. And you see, all these things together start to wonder whether this model of mutation, accumulation of mutation cancer is correct. For example, this graph here is very nice. Shows, you know, that if you have a tissue with the high stem cell division, you have more risk of cancer than a tissue like the bone with low level. True. But now let's take again the different tissues with different capability, and let's see not the total amount of cancers, but the age dependence of the cancers. Well, if you normalize, you see, of course, they look different, but if you because of course, some cancers, like this is the colon cancer, and this is pancreas cancer. But if now, if now you normalize the absolute value, you know, max half zero, the age dependence is almost the same. Almost the same. There is always, you see, a sort of small increase here, which is essentially driven by strongly genetic dependent tumors that are pediatric tumors. Typically, you see this is seen in leukemia, especially acute myeloid leukemia, the AML. But then you have a very low value until you pass through 50, 60, and then you really go very much up. So cancer is a disease of the aging. And interestingly enough, we call aging after 50 years now. But if you look at the old times, if you look at the survival course in Europe 15,000 years ago, this was the survival curve. So the fraction of people reaching more than 60 years was totally negligible. Most of the people were dying around 30, 40 years, and they were dying mostly for infections. Let's say there was no control on infection. There was a large neonatal death, and then of course there was a large death for other causes. So at that time, for the genetics of the humans, let's say, cancer is a rare disease, very rare disease. Now is the second cause of death worldwide, but at that time was a very rare disease. So we didn't adapt, because you see, this is now the curve of cancer. You see, the great peak is here between 60 and 80 years old, but at that time there was nobody with that age. And that's why there is a nice book now from James De Gregori, who is professor of oncology at the University of Colorado. It's called Adaptive Oncogenesis, where the essential hypothesis is that that cancer is an evolutionary program, and is the lack of pressure, the lack of control that we have after a certain age on the proliferation of the cell that cause cancer. We develop genetically, we are able to control cancer when we are young, because that's when we are supposed to be alive. But down here, where we are not supposed to be alive, we are alive only now thanks to medicine, thanks to the improvement in technology, then genetically we are not prepared to control our tumors. I think it's fascinating. And it's related somehow to the regenerative capacity. You see, these are different animals with different regenerative capacity. For example, these are lizards, you know, very strong regenerative capacity, no cancer, and so forth and so forth. So essentially, when an organism has an efficient regenerative mechanisms, like it can be a lizard or it can be a young man, there is no cancer. When you get aging or is affected by noxious agents, I mean smoking and so on, then you decline your regenerative ability, and injury causing loss of cells or diminished cellular function cannot be adequately compensated by cellular division. And that's what causes cancer. So it's not, you see, the normal cell division can compensate the development of cancer in young age, but not in late age. This is now the statistics. We have seen already in the plot for the stem cells what are the most common cancers, but of course this depends on the habits. These are United States in 2019. This is a paper that is published every year on a journal called Cancer Journal for Clinicians. In 2019, these are the new cases. So if you want, this is the incidence of cancer, and this is the mortality. You see, it's not exactly the same. For example, for women, the first cancer is breast, followed by lung, colon, and uterine. But in terms of mortality, you see, lung is worse than breast. Breast is less lethal than lung. Lung is a very lethal tumor. I mean, still today, you have about only 20% survival for lung cancer. Breast is now something like 50% survival, if not more. All depends on the stage, of course. So I say in average, we will see that there are many stages of cancer. For men, you see in terms of incidence, you have prostate at the first, then lung, colon, and urinary bladder. In mortality, first is lung again, second is prostate, and then colon, pancreas. Just want to say that it is significant that lung used to be by far the first, also still the first in mortality, used to be by far the first also in incidence. Now is going down, has been surpassed by prostate in men. Is still pretty high, you see, still pretty high in females, because actually apparently the amount of reduction in smoking, to the point that, you know, lung is the first mortal in women, the reduction in smoking is not enough compensated by in women as compared to men. So there is more people quitting smoking among men than girls, boys and girls, because this is really in the young age. You also see something interesting. There are some tumors like pancreas that are pretty low in incidence. You see, they are down here, but they are pretty high in mortality. And you see why. They expect 30,000 new cases, and they expect 24,000 deaths. So the mortality is almost the same as the the incidence. This is, of course, a big tragedy. Some tumors, like pancreas cancers, are really still incurable. This hypothesis, the cancer stem cells hypothesis, I think we saw already. This is just a reminder where there is more stress on the cancer stem cell concept. So we saw that you need a mutation, and then this mutation will lead to a tumor. But at the end, what happens? You see that you have a stem cell, and then from these stem cells accumulating mutation, you have cancer. This can be in the stem cells or can be in the progenitor cells. You remember, progenitor cells are after the stem cells. What is interesting is that the stem cells are the cause of cancer, but then eventually the cancer stem cells are those that make the cancer grow. And so when there is a therapy against cancer, it's very important that this therapy is able to destroy the cancer stem cells. Because you see, if you destroy the cells but not the cancer stem cells, you will have a regrowth. But if you are able to destroy the cancer stem cells, then the tumor will be stopped. Of course, the situation is more complicated because there is not only cancer stem cells and cancer cells. We have seen there are many other factors, like these are fibroblasts, for example, here. So there can be situations where you can kill the cancer stem cells, but then you have a relapse because some cancer cell is reverted into a cancer stem cells. And this can be due to the microenvironment. This was the last slide with a little joke here for you. And I know it was a long lecture, this one, but I hope it was useful. And I will be happy to answer any questions on Wednesday related to this lecture. Thank you.